Hello students, let's start the class. It's exactly 10 o'clock. So all of you will join till date. Let's recall what we have done yesterday. तो हमने कल क्या पढ़ा था उसके बारे में एक बार डिस्कस करते हैं. हमने बेसिकली कल सेंट्रल डॉगमा पढ़ा था ऑफ मॉलिक्यूलर बायोलॉजी. हम अभी सेल बायोलॉजी पढ़ रहे हैं, बट सेल में कुछ मॉलिक्यूलर लेवल पे कुछ टर्म्स जो हमें पता होनी चाहिए वो हम जानने के लिए हमने सेंट्रल डॉगमा की बात की थी. सेंट्रल डॉगमा ऑफ मॉलिक्यूलर बायोलॉजी. अकॉर्डिंग टू सेंट्रल डॉगमा ऑफ मॉलिक्यूलर बायोलॉजी, डीएनए, अब ये डीएनए कहाँ है हमारा? सेल में. सो हम सेल के अंदर डीएनए की बात करें तो इसकी कुछ टर्म्स के लिए हमने यहाँ पे बात की थी कि सेल प्रेजेंट इन द न्यूक्लियस ऑफ अ डीएनए हैव इंडिपेंडेंट सिंथेसिस. मींस डीएनए कैन मल्टीप्लाई इटसेल्फ और अपना कॉपी नंबर इंक्रीज कर सकता है. डीएनए डिपेंडेंट डीएनए सिंथेसिस इज कॉल्ड डीएनए रेप्लिकेशन. क्लियर? एंड फ्रॉम डीएनए mRNA, messenger RNA is formed, that is called transcription. Clear? And from mRNA proteins are formed, that is called translation. So हमें यहाँ पे बेसिकली अभी क्या लर्न करना है? These terms. What is replication? DNA dependent DNA synthesis, and from DNA RNA is formed, that is transcription, and from RNA proteins are formed, that is translation. Clear? Now this DNA dependent DNA synthesis, that is replication and transcription, occur in nucleus. ये सारा वर्क कहाँ हो रहा है? न्यूक्लियस में. Clear? And this translation occurs in ribosomes, and ribosomes are present in cytoplasm. साइटोप्लाज्म, और साइटोप्लाज्म में भी कहाँ? राइबोसोम्स. तो हमने एक्चुअली क्या पढ़ा था कि राइबोसोम्स आर द साइट ऑफ प्रोटीन सिंथेसिस. आई थिंक आप सबने अभी तक अटेंडेंस मार्क कर दी होगी अपनी. मार्क योर अटेंडेंस इन द कमेंट सेक्शन, एंड इफ यू हैव एनी डाउट, राइट योर डाउट इन अ कमेंट सेक्शन सो दैट आई विल क्लियर योर डाउट. नाउ अभी तक हमने क्या पढ़ा है सेल चैप्टर में? द इंट्रोडक्शन ऑफ अ सेल, डिस्कवरी ऑफ द सेल, एंड इन लास्ट क्लास वी डिस्कस्ड अबाउट द प्रोकैरियोटिक सेल. हमने बैक्टीरियल सेल के स्ट्रक्चर के बारे में डिस्कस किया था. हाउ इज द सेल एनवेलप ऑफ द बैक्टीरियल सेल, टाइप्स ऑफ बैक्टीरियल सेल्स, तो वो सब डिस्कस कर लिया. आज हमने स्टार्ट करना है यूकैरियोटिक सेल से. सो लेट्स स्टार्ट टुडेज क्लास. Today we will start with eukaryotic cell. यूकैरियोटिक सेल की बात कर रहे हैं तो सबसे पहले हमें पता है कि हमारे माइंड में स्ट्राइक करेगा एनिमल सेल एंड प्लांट सेल आर द यूकैरियोटिक सेल. सो फर्स्टली लेट्स ड्रॉ द डायग्राम ऑफ दीज यूकैरियोटिक सेल्स. आई एम गोइंग टू ड्रॉ द डायग्राम ऑफ एनिमल सेल फर्स्ट. दिस इज एनिमल सेल. Reducing the size because I think board is not visible from the lower side, that's why I am drawing in small manner. So the outermost membrane of the animal cell is plasma membrane. Clear? And in center it has nucleus. Nucleus has nuclear membrane. This nuclear membrane is double membrane bound. This nuclear membrane is double membrane bound with nuclear pores. These are nuclear pores. So see this is nuclear membrane, and this nuclear membrane is double membrane bound. It is double layer membrane and it is not continuous. It has some pores. What is the function of these nuclear pores? अभी हमने पढ़ा कि डीएनए से RNA, that is transcription, inside the nucleus होगी, and that RNA moves in the cytoplasm and undergoes the translation that is protein synthesis. तो RNA को जब मूव करना है तो वो किसके थ्रू मूव करेगा? थ्रू न्यूक्लियर पोर. Clear? These are nuclear pore, and the fluid, jelly-like fluid present inside the nucleus is called nucleoplasm, and it has RNA and protein-rich nucleolus and DNA as genetic material. अब जब डीएनए एज अ जेनेटिक मटेरियल की बात कर रहे हैं तो डीएनए किस फॉर्म में प्रेजेंट होगा? क्योंकि ये यूकैरियोटिक सेल है, हमने डिफरेंस में पढ़ा है कि प्रोकैरियोटिक सेल में डीएनए इज प्रेजेंट इन द फॉर्म ऑफ सिंगल सर्कुलर डीएनए, बट इन यूकैरियोट्स इट इज प्रेजेंट इन द फॉर्म ऑफ क्रोमोसोम्स. क्लियर? बट अभी मैं यहाँ क्रोमोसोम्स नहीं बना रही हूँ, आई एम हियर ड्रॉइंग द क्रोमेटिन मटेरियल. व्हाट इज द डिफरेंस बिटवीन? वी विल डिस्कस इन डिटेल when we'll study about the nucleus. Okay, now this is nucleus and this is the fluid present in the whole cell is cytoplasm. अच्छा, यहाँ एक टर्म क्लियर करते हैं. हमने एक टर्म पढ़ी है प्रोटोप्लाज्म. क्लियर? प्रोटोप्लाज्म के बारे में हमने पढ़ा है. व्हाट इज प्रोटोप्लाज्म? प्रोटोप्लाज्म इज द लिविंग फ्लूइड प्रेजेंट इनसाइड द सेल. मींस प्लाज्मा मेम्ब्रेन के अंदर जितना भी लिविंग कंटेंट है, चाहे वो न्यूक्लियस हो, न्यूक्लियस का न्यूक्लियोप्लाज्म हो, और द सेल ऑर्गेनेलीज और द साइटोप्लाज्म. सो हम वी कैन से साइटोप्लाज्म प्लस न्यूक्लियोप्लाज्म. क्लियर? This nucleoplasm, the whole content in this cytoplasm also contains cell organelles. So this total is protoplasm. इसके बाद फिर हम एक और टर्म पढ़ते हैं cytosol. Now what is this cytosol? Cytosol is the fluid present except cell organelle. मीन्स जब हम साइटोप्लाज्म की बात कर रहे हैं तो हम क्या कह रहे हैं? द जेली लाइक फ्लूइड प्लस द सेल ऑर्गेनेलीज लाइंग इन दैट साइटोप्लाज्म और जेली लाइक फ्लूइड. बट साइटोसोल मीन्स द इंटरसेलुलर फ्लूइड कह सकते हैं हम इसे. इंटरसेलुलर और इंटरसेल ऑर्गेनेली फ्लूइड प्रेजेंट इन बिटवीन द सेल ऑर्गेनेली मीन्स वो साइटोप्लाज्म का वो पोर्शन जिसमें सेल ऑर्गेनेलीज काउंट नहीं हो रहे, सिर्फ दैट जेली फ्लूइड काउंट हो रहा है. क्लियर? नाउ द थर्ड मोस्ट थिंग इज, हमने पढ़ा है बेसिक ऑर्गेनाइजेशन ऑफ सेल में आउटर मोस्ट मेम्ब्रेन इज प्लाज्मा मेम्ब्रेन, देन न्यूक्लियस, and in cytoplasm cell organelles are there. So now we will talk about the cell organelles. First cell organelle is endoplasmic reticulum. These are tubular structures that are in continuation with nucleus. These are the tubular structures present like this. Clear? Now these ribosomes are of two types. These are endoplasmic reticulum are of two types. One with ribosomes. One with ribosomes are known as rough endoplasmic reticulum. जिन एंडोप्लाज्मिक रेटिकुलम के सरफेस पे राइबोसोम्स होंगे हम उन्हें क्या बोलेंगे? रफ एंडोप्लाज्मिक रेटिकुलम. एंड द राइबोसोम्स आर नॉट प्रेजेंट ऑन सम एंडोप्लाज्मिक रेटिकुलम, दे आर नोन एज स्मूथ एंडोप्लाज्मिक रेटिकुलम. सो दीज आर rough endoplasmic reticulum, and on these rough endoplasmic reticulum ribosomes are present, and these ribosomes are 80s. हमने पढ़ा है डिफरेंस में कि in prokaryotic cell 70s ribosomes are there, while in eukaryotic cell 80s ribosomes are there. And then next to this, in the very close to nucleus, these Golgi bodies are present. These are Golgi bodies. Clear? And the most important cell organelle, mitochondria. It is double membrane bound cell organelle. This is mitochondria, known as powerhouse of the cell that releases energy in the form of ATP. हमारी बॉडी में जितनी भी एनर्जी रिलीज हो रही है वो कहाँ हो रही है? माइटोकॉन्ड्रिया में, बाय ऑक्सीडेशन ऑफ फूड. Clear? And one more thing is there, that is this is centrosome. This is centrosome. Centrosome contains, this centrosome has two centrioles present perpendicularly to each other that helps in cell division, and this is a cell organelle that is present only in animal cell. ये और कहीं present नहीं है, plant cell में नहीं है, fungal cell में नहीं है. So lysosomes. Next is lysosomes. Lysosome, small structure that contains enzyme and helps in digestion of foreign particle. तो हमने जो last cell organelle से पढ़ी, that is lysosomes. Lysosomes contain enzymes. Clear? This is the diagram of animal cell. जिसमें हमने क्या पढ़ा कि it is outermost membrane, plasma membrane, then nucleus, and then cell organelles, endoplasmic reticulum, Golgi bodies, mitochondria, lysosomes, and some small vacuoles are also present. These vacuoles may or may not be present. In some of the bacterial, some of the animal cell vacuoles are present, very small vacuoles are present, and some other they are not present. That depends on the function of that animal cell. So this is about the animal cell. Now I will draw the plant cell. Now I will draw eukaryotic cell, that is plant cell. We will majorly focus on the differences between the animal cell and plant cell here. So outermost cell wall is present, and this cell wall is made up of cellulose. In the cell wall is a dead cell wall because it is a made up of cellulose. Cellulose की बनी और ये dead है. Dead होने की वजह से clear. This is cell wall, and in cell wall pits and middle lamella are present. Brain not have cell wall, while in animals covers the 70% of the volume of a plant cell, and this vacuole is called sap vacuole. Why it is called sap vacuole? Because it is filled with water and other nutritive fluid, and the membrane of this sap vacuole is known as tonoplast. This is tonoplast membrane of vacuole. Now because of presence of this large vacuole, the nucleus is shifted towards the corner, periphery. Animal cell में हमने क्या देखा था? न्यूक्लियस थोड़ा साइड में. Now I will draw the nucleus. Nucleus is same in structure, having nuclear membrane, double nuclear pores, double membrane nuclear, and nucleolus and chromatin as a gel. So I am labeling it here as only nucleus because हमने इसके detail parts animal cell में देख लिए, वो बिल्कुल same है. Clear? Again now endoplasmic reticulum. It has continuous endoplasmic reticulums. Reticulums are of two types: smooth endoplasmic reticulum, and if they have ribosomes, they are rough endoplasmic reticulum, and ribosomes are ATS type, and some of the ribosomes also may lie freely in the cytoplasm. Then is Golgi bodies. These are Golgi bodies. Now these are Golgi bodies or Golgi apparatus we can say, and in plant cell sometimes they are also known as dictyosomes. They are also called dictyosomes. And now the mitochondria. This is mitochondria that provide energy by oxidation of food in the form of energy in the form of ATP. And here I am drawing chloroplast. These are the chloroplast. This is another double membrane bound cell organelle. Clear? This is not present in a animal cell. Now vacuoles. It also has, this has vacuoles, lysosomes. These are lysosomes, and lysosomes in plant cells are also known as saprosomes. They are also known as saprosome. So हमने क्या differences पढ़े? The main difference is presence of cell wall, one large vacuole, and presence of chloroplast, and nucleus is shifted towards the corner. And these are same. These are the only difference in the name only. In Golgi bodies, in plant cell is also known as dictyosomes, and lysosomes are also known as plasma membrane. Now we will study about the plasma membrane in detail. We will study the structure of plasma membrane. So we have done with the diagram of animal cell and plant cell. Now we will start the detailed structure. So we will start with the structure of plasma membrane. Structure of plasma membrane. Plasma membrane, first of all, it is the outermost membrane present in a cell that allows some selected molecules to enter or exit the cell. Means this plasma membrane, that is the outermost membrane that covers the cell, is selectively permeable membrane. Selectively permeable membrane that allows some selected molecules to enter or exit the cell. I will give you one example. When you apply soap on your skin during bathing, it is generally absorbed, but water is not absorbed when you bath. Water is not absorbed by the skin cells because it is selectively permeable. It does not allow water to enter. While when you put gram seeds or kidney bean seeds in a water is easily absorbed. So again, the membrane of seeds is such a selectively that it absorbs water. When you apply cream on your skin, it is absorbed, but water do not. So the plasma membrane is selectively permeable membrane. It allows some selected molecules to enter or exit the cell. अब हम स्ट्रक्चर की बात करते हैं. तो स्ट्रक्चर को किसने दिया था? फर्स्ट स्ट्रक्चर, द मॉडल ऑफ द स्ट्रक्चर वाज गिवन बाय Davison and Danieli, and they give the structure name sandwich model. Sandwich means a two layer of bread filled with some material. So ऐसा ही कुछ हम यहाँ बनाएंगे. Now according to them, plasma membrane is made up of phospholipid bilayer. This is phospholipid bilayer in which the two lipid layers are facing. These proteins are globular protein, not fibrous protein. It is globular protein, and this is phospholipid, and this is again protein. So sandwich बन गया ये. The two protein layer covers the phospholipid bilayer. Now, according to Davison and Danieli, the thickness of these protein is 20 angstrom on both the side, and 35 angstrom is the thickness of the phospholipid bilayer. So the total thickness of the plasma membrane is approximately 75 angstrom. Clear? And these phospholipid bilayer has, and this bilayer is, lipid bilayer is hydrophilic. क्या हो गया? एम्फी एंड अनदर इज हाइड्रो इज हमें पाए वो सिलेक्ट सॉल्युबल हो उसको भी पास करने देगी बट अकॉर्डिंग टू दिस स्ट्रक्चर from here fat soluble molecules can easily pass but water soluble or water cannot easily pass because this hydrophilic lipid bilayer repels the water molecules and does not allow it to pass. So this is a limitation of this plasma membrane model, that is sandwich model given by the Davison and Renai. So this is not accepted. Clear? Now after that one another model came, that is unit membrane model. Next is structure of plasma membrane. Second one is unit membrane model given by Robertson, and according to him this is not a 3 layers, these are not 3 layers. उसने जो स्ट्रक्चर एक्सप्लेन किया रॉबर्टसन ने प्लाज्मा मेम्ब्रेन का वो एग्जैक्टली सेम था. उसने बोला कि ग्लोबुलर प्रोटीन्स है, फॉस्फोलिपिड बायलेयर है जिसमें हाइड्रोफिलिक एंड हाइड्रोफोबिक पार्ट है. सो दिस इज हाइड्रोफोबिक एक्चुअली corrected. This is in the sandwich model also. This is hydrophobic, water hating, and this is water loving. तभी उसने water को पास नहीं होने दिया था. तो same is the structure. बस उसने एक बात कही कि these three are not different layers. We cannot consider as different layers. This is only one layer of 75 angstrom. Clear? Widely accepted model था ये at that time. But अभी भी हमारा जो concern था कि fat molecules from this membrane, but what about the water soluble molecules? वो किस मॉडल? बिफोर दैट हमने क्या पढ़ा? सैंडविच मॉडल एंड यूनिट मेम्ब्रेन मॉडल की एक सेम एक्सेप्शन थी और जो चीज एक्सप्लेन नहीं कर पाए थे, मूवमेंट अक्रॉस द प्लाज्मा मेम्ब्रेन, मूवमेंट ऑफ वाटर सॉल्युबल मॉलिक्यूल्स अक्रॉस द प्लाज्मा मेम्ब्रेन, वो एक्सप्लेन नहीं कर पाए थे. एंड नाउ आफ्टर दैट इज फ्लूइड मोज़ैक मॉडल. Third is fluid mosaic model. This is most widely accepted model. आपका ये नीट में क्वेश्चन आया हुआ है कि which of the following is the most widely accepted model of the plasma membrane? So it is fluid mosaic model given by Singer and Nicholson in 1972. This is again very important. Singer and Nicholson gave the fluid mosaic model of the plasma membrane, that is widely accepted model, in a 1972. Now this is widely accepted model, and the possibility of this model is just because of the discovery of electron microscope. This was given only after the discovery of electron microscope. Again, according to Singer and Nicholson, plasma membrane is made up of phospholipid bilayer, that is amphipathic in nature, and proteins, but the arrangement is quite different. तो इसने क्या बोला सिंगर निकोल्सन ने कि this is phospholipid bilayer clear. This is phospholipid bilayer. Just because of that, it is amphipathic. Proteins हैं, two types of proteins. One is extrinsic protein and another is intrinsic protein. Here on the surface also protein molecules are present on the outer surface, and the proteins that are embedded in a phospholipid bilayer are and some proteins are embedded either fully or partially. Some proteins are only partially embedded. These are intrinsic protein. Now, the movement across the plasma membrane is of two types, and that is possible just because of these proteins. Clear? So, we will talk about now theory, and another is flip-flop movement of phospholipid bilayer. See, when these phospholipid move like this from this first, second, third and fourth, first within lipid bilayer. Within bilayer, lipid bilayer, this is transition movement. But flip-flop movement means within two layers, two different layers. So plasma membrane shows phospholipid bilayer of a plasma membrane shows movement, and that is transition type when it is occurring within a lipid layer, or if it occurs between the two layers, then it is flip-flop movement. And these extrinsic proteins also shows the movement, and due to that movement permeable. Now see how the movement occurs. Movement occurs and the transport occurs mainly. Now these fat soluble molecules can easily pass because of the nature. Clear? Because these extrinsic proteins also show the some movement, lose movement, जिसकी वजह से fat molecules easily pass out हो जाएंगे. And now about the water soluble. So water soluble molecules can pass through these proteins. So these proteins are also known as portions, or we can say channel proteins. Channel proteins क्यों? क्योंकि ये soluble molecules को pass out होने दे रहे हैं. portions, और स्पेशली अगर वो सिर्फ वाटर को पास होने दे रहे हैं, वाटर सॉल्युबल सब्सटेंसेस, प्योर वाटर अगर पास हो रहा है, तो इट इज कॉल्ड एक्वापोरियन, एंड दे आर आल्सो नोन एज चैनल प्रोटीन. नाउ वन line is given by the fluid mosaic model. For the fluid mosaic model, for the that phrase for given by the Singer and Nicholson is proteins are icebergs in sea of lipid. Means these proteins are just floating because of their light weight. जैसे कि आइसबर्ग कंपेयर किया है प्रोटीन्स को आइसबर्ग से और सी को लिपिड से, कि लिपिड्स जो हैं वो आइसबर्ग्स की तरह लिपिड्स में प्रोटीन्स फ्लोट कर रहे हैं, तो जिसकी वजह से उसका नेचर क्या हो जाएगा? फ्लूइडी नेचर, एंड दैट्स व्हाई इट इज कॉल्ड क्वासी हाफ सेमी फ्लूइड नेचर, or we can say fluid mosaic. Fluid means plasma membrane has capability for the movement, that's why fluidy. But mosaic means false. Truly mosaic fluid नहीं है because of the presence of these proteins and bilayer. हम इसको बिल्कुल fluid नहीं कह सकते, but it is quasi fluid because it is just like the proteins are iceberg in a sea of lipids. So this is all about the fluid mosaic model. Now we will discuss about the different types of movements transport across the plasma membrane through the fluid mosaic model, जिसकी वजह से ये quasi fluid nature है. Write your doubts in a comment section so that we can discuss the doubts also. Now across membrane, transport across membrane. For the two transports, there are two types of transport. One is passive and another is active transport. Passive transport means do not require energy, ATP not required. First point. For the active transport, energy is required, that is ATP required. This is the first difference. And the second, the most important difference is movement occur across concentration gradient. Low concentration. कब तक? टिल द इक्विलिब्रियम इज अटेन, मीन्स जब तक दोनों साइड पे कंसेंट्रेशन सेम नहीं हो रही है. तो मूवमेंट फ्रॉम हाई टू लो इज कंसेंट्रेशन ग्रेडियंट. हेयर ऑपोजिट टू concentration, opposite to concentration gradient means the molecules move from low concentration to high concentration. ये जो gradient है उसमें ज्यादा energy require होगी, that's why it is called active transport is called uphill transport. Now this passive transport where no ATP required and the movement occur across the concentration gradient, it is further of three types. One is सिंपल सिस्टम. हम ये नहीं कह सकते कि डिफ्यूजन सिर्फ लिविंग ऑर्गेनिज्म में होती है, इट ऑल्सो अकर इन नॉन लिविंग सिस्टम. ऑल्सो सिंपल डिफ्यूजन में यहाँ पे हमें कोई फैसिलिटेटेड डिफ्यूजन, मूवमेंट ऑफ मॉलिक्यूल्स फ्रॉम हाई कंसंट्रेशन टू लो कंसंट्रेशन थ्रू पोरियोन्स. पोरियोन्स अभी हमने पढ़े थे, द प्रोटीन मॉलिक्यूल्स, जो हमने अभी सिंगर निकोल्सन के फ्लूइड मोज़ैक मॉडल में इंट्रिंसिक प्रोटीन्स की बात की. चैनल प्रोटीन, व्हेन ट्रांसपोर्ट अकर थ्रू दैट चैनल प्रोटीन, दैट इज कॉल्ड फैसिलिटेटेड डिफ्यूजन. एंड all the water soluble substances always show facilitated diffusion. Then the last, osmosis, the movement of water molecules, or you can say solvent molecules, only from high concentration to low concentration through movement of solvent. First of all, from high concentration to low concentration following the concentration gradient, and next is through semi permeable membrane, and this plasma membrane is semi permeable, means ये सिर्फ कहाँ होगी, लिविंग सिस्टम में. ठीक है. और yes, we can do osmosis artificially also, but for that we need to make this artificial semi permeable membrane, and it is generally made from the animal skin. Clear? So there are the two types of transport, that is one is passive transport and another is active ट्रांसपोर्ट. तो आज हमने यूकैरियोटिक सेल में डिटेल स्ट्रक्चर की बात की अबाउट द प्लाज्मा मेम्ब्रेन. टुमारो वी विल स्टडी अबाउट द सेल ऑर्गेनेलिस. दैट, थैंक यू.